cord. Okay. So at the metaphase 1, the homologous chromosomes or the bivalent, we are still calling them bivalent because they are still attached to each other at the level of the chiasma. Whenever this chiasma attaching those homologous chromosomes together disappear, we won't call them bivalent or tetrad anymore, okay? So those homologous chromosomes will attach to the microtubule spindle by their kinetochore. As you know, do you know what is the kinetochore? Okay, you don't know. The kinetochore is a protein complex that bind to the centromere region of the chromosomes. هيدا in blue. Okay, here we have the centromere, and in blue we have the kinetochore, and this protein complex will bind to the mitotic spindle or to the microtubules during the metaphase. So is the presence of this kinetochore will allow for the chromosomes to bind to this spindle, okay, during the metaphase or the prometaphase. So we need the presence of the kinetochore for the attachment of the chromosomes on the microtubule spindle. Now, the homologous chromosomes will attach via their kinetochore to the microtubules. Okay, as you can see, they are attached to the microtubule. Where do they attach during the metaphase?
An equatorial plate.
Yes, they will line up on the equatorial plate, and in the middle of the cell. Like here, they are in the middle of the cell. Now, as you know, they are still attached together. The red and the blue chromosomes, there are homologous chromosomes. They are still attached at the level of chiasma. Okay, so how will they bind to the microtubule spindle? Yani, does all the, let's say, paternal chromosomes will face the lower pole of the cell and all the maternal chromosome will face the upper pole, or the inverse, or how will they do? In fact, their orientation on the microtubule spindle is random. So there is no rule. Yani it can either be the maternal chromosome facing the upper side or the upper pole of the cell, or the lower pole of the cell. Their orientation on the mitotic spindle, or sorry, on the microtubule spindle is random, okay? So either homologue, either chromosome of the pair can be oriented to either pole of the cell. Okay, and this orientation, this random orientation, will allow an increase in the variety or in the diversity of genetic material in the daughter cells. We will see how in the anaphase, okay? So the orientation of chromosomes on the spindle is random, which means the arrangement of the paired chromosomes with respect to poles of the spindle is random along the metaphase plate. Each pair may orient with either its maternal or paternal homologue closer to one pole. مش يعني انه كل الباترنال مبرومين لفوق والماترنال لتحت، عشان لما تصير الديفيجن الماترنال بينزلوا كلهم على one cell والباترنال بيطلع على one cell. لا. They are randomly organized on the equatorial plate. So the pair of homologous chromosomes are randomly oriented at metaphase of meiosis 1. During the anaphase, as you know, the homologous chromosome, the anaphase 1, the anaphase of the meiosis 1, we will have here the separation of homologous chromosomes. Okay, be careful, not of the chromatids, but of the homologous chromosomes. And they will separate and segregate independently, yani randomly, okay? As we said, because they were oriented randomly on the microtubule spindle, here they will get segregated independently. So but just before their separation, we should have the dissociation of the chiasma. So as we have seen, the chiasma will remain present until the anaphase, and it is at the anaphase 1 stage that the chiasma will disappear, will get dissociated to allow the separation of the homologous chromosomes. Okay. And the sister chromatids of one chromosome will remain attached to each other, okay, at the level of the centromere. So the centromeres are not cleaved to keep on having one entire chromosome on each pole. Okay, we don't need to separate the chromatids now. We need to separate the homologous chromosomes. That's why here the centromere is maintained. The sister chromatids are still maintained attached together at the level of the centromere. And each homologous chromosome is pulled toward a pole of the cell. Each homologous chromosome will be pulled toward a pole of the cell by the microtubules. Now, the microtubules which will bind the kinetochore are called the kinetochore microtubules. Okay, so each homologous chromosome will be pulled to a pole of the cell due to the kinetochore microtubules. Because as we have said, the chromosomes, the homologous chromosomes, are attached to the spindle by the kinetochore. Okay, so the kinetochore is binding the microtubule, and they will be pulled toward a pole by those microtubules attaching to the kinetochore. We call them the kinetochore microtubules. And now, what happens due to this separation of the homologous chromosomes? We'll have, as you can see, some blue chromosomes on the upper side of the cell and red chromosomes, and also some blue and red chromosome on the lower side. So this is also a mixing of, or a segregation of the chromosomes and of the genetic material, because when the cell will divide, okay, on each pole we will have some combination or composition of the chromosomes, some maternal, some paternal, and the same way as on the lower pole. And in a different cell, we will have a different segregation depending on how the chromosomes were attached to the microtubule spindle. Okay, so this random orientation of those chromosomes or of those homologous chromosomes on the microtubule spindle will allow their independent segregation and will allow an interchromosomal shuffling, which means we call it an interchromosomal mixing, okay, or assortment. We will have an independent assortment of chromosomes. So we'll have a random assortment of the chromosomes. Let's say in one cell we will have the chromosome 1 and 3 will be maternal on the upper pole, and the chromosome 2 and 4 will be paternal. And in another cell, no, we will have 1 and 3, let's say paternal, and 2 maybe paternal also, but 4 maternal. So we will have a different genetic composition, a different assortment of the chromosomes on each pole of the cell. And this will create a higher diversity in the offspring, in the gametes and in the offsprings. And that's what we call the independent assortment of chromosomes, or the interchromosomal shuffling. Okay, so we have a mixing between the chromosomes. This is the interchromosomal shuffling, okay, or independent assortment of chromosomes. Is it clear?
دكتور
Yes.
دكتور كينوتوكور مايكروتيوبيولز ما فهمت كتير فكرتها، وين موجودين أو شو بيعملوا؟
No, kinetochore microtubules, they are هن كلهم هول microtubules, okay? So we have those microtubules that bind to the chromosomes. They are called the kinetochore microtubules. بس هيك.
أوكي.
اوكي يعني بس بنسميهم kinetochore microtubules. Now at the telophase we will have we arrived here at the metaphase, at the anaphase, and now at the telophase we will have the cytokinesis. We will have also the formation of the nuclear envelope. Okay, so the nuclear envelope will start to form again, and by the end of the telophase it will be complete. And we will have a cytokinesis, which will allow the division of the parent cell into two daughter cells. And we will have two different cells, two daughter cells, each one having one set of chromosomes. So they will have 23 chromosomes instead of 46. So we will have two haploid daughter cells, each one having two chromatids. Okay. Now, so here we can review the different stages of the meiosis 1. We have the prophase 1, where we had the crossing over, the binding of the homologous chromosomes at the level of the chiasmata. At the metaphase 1, they will associate or attach to the microtubules and on the equatorial plate, and their orientation is random on this equatorial plate. At the anaphase, we'll have detachment of the chiasmata and separation of the homologous chromosomes, each one to a pole of the cell. At the telophase, we have the regeneration of the nuclear envelope and the cytokinesis, the separation of the two daughter cells, and we'll have one haploid cell, two daughter cells, which are haploid and having two sister chromatids. Now, the meiosis 2 is like a normal mitotic division as we have seen. نعم.
بس متى حنخلص الميتينج؟
يلا بنخلص هيدا الميوسيس 2 وبنخلص. اوكي. It's like عندكم شي بعد هاي السيشن؟
لا.
Okay. The meiosis 2 is like a normal meiotic division. We will have during this meiosis a separation of sister chromatids. So the number of chromosomes will remain the same as in the meiosis 1. At the end of the meiosis 1 كان عنا 23 chromosomes, so one set of chromosomes. Here at the end of the meiosis 2, we will conserve the same number of chromosomes. That's why it is called an equational division. And we will still have an equal number of chromosomes as the end of the meiosis 1, but it's the number of chromatids that will be reduced, and we will have cells with one chromatid. At the prophase 2, the nuclear envelope will break down, and we will have the appearance of a new spindle of microtubules that will form. Okay. At the metaphase 2, also the chromosomes will bind by their kinetochores to the mitotic spindle. Here we are seeing the spindle fibers from opposite poles bind to kinetochore of each sister chromatid. Because look, we have kinetochores on each chromatid, okay? So now in the meiosis 2, the kinetochores of each chromatid will bind to the microtubule spindle. In this way, whenever this microtubule spindle will contract, okay, each chromatid will get separated to a pole, okay? And we have, at the anaphase, we have the cohesin proteins that are maintaining those two chromatids attached together at the level of the centromere, they will be degraded. Okay, and when we will have the microtubule spindle that will pull those two or that will contract, okay, the two chromatids will be pulled to each one to a different pole of the cell. So in the metaphase, we have the spindle fibers from opposite poles bind to kinetochores of each sister chromatid, because as we said, the kinetochores are present on the centromere region of each chromatid. Okay, and those kinetochores of the two chromatids, both of them are binding the spindle. And then they will migrate to the equatorial plate. They will be at the equatorial plate. At the anaphase 1, each chromatid is pulled apart to opposite poles. And the coenzyme complex, why it is made possible, why we can pull them apart and separate them, because the coenzyme complex maintaining or joining the centromeres of sister chromatids will be destroyed. We don't have any more hydal protein complex that is joining the chromatids at the centromere region. This coenzyme complex will be destroyed, so we can separate the two different chromatids. So the centromeres are splitted and the sister chromatids pulled to opposite poles. So here each, هيدا in the anaphase 1 here. The anaphase, the metaphase 2, we will have the chromosomes on the equatorial plate, and then during the anaphase, each chromatid will be pulled to one pole of the cell. And at the telophase, we will have the nuclear envelope that will reform again around the chromosomes of each daughter cell. And we will have the cytokinesis on each cell that will divide each cell to two daughter cells. And as a result, we will have 4 daughter cells, 4 haploid daughter cells, each one having 1 chromatid. Okay. So here how the sister chromatids or each chromosome is binding the microtubule spindle by their kinetochore. In yellow, you have the kinetochores. Okay. So whenever we have this contraction of the at the anaphase, you will have the coenzyme complex joining the chromatids together that will be destroyed. Okay. And the centromere will be splitted, and each chromatid will go to a different pole, and that's how we can separate them in 2 different cells. Okay. Now, what is the difference between the mitosis and the meiosis? In both of them, the mitosis and the meiosis, as we said, we have a DNA replication that happens just before, in the interphase, before the cell enter in mitosis and before its entry in meiosis. Okay, but the DNA replication in the meiosis occurs only before the meiosis 1, not before the meiosis 2. The number of divisions in the mitosis, we have 1 division, which has the prophase, metaphase, anaphase, and telophase. But in the meiosis, we have 2 divisions. We have the meiosis 1 and the meiosis 2. Each one of them includes a prophase, metaphase, anaphase, and telophase. In the mitosis, we don't have a synapses between the chromosomes, so the homologous chromosomes don't attach to each other or don't get held to each other in the mitosis. However, in the meiosis, we have a synapses only during the prophase 1. Okay, we have a synapses during the prophase 1 with crossing over between non-sister chromatids. The number of daughter cells in the mitosis, we generate 2 daughter cells, which are diploid, having 2 N chromosomes, 2 sets of chromosomes, and the daughter cells are completely identical to the parent cells, okay, because they still have the same genetic material. However, in the meiosis, we generate 4 daughter cells, not 2. They are haploid cells, and the daughter cells are genetically different from the parent cell and from each other. Okay, do you know why they are different from each other?
من خلال الكروسنج اوفر اللي صار والميكسينج
Yes, من خلال crossing over وال independent assortment أو اللي قلنا إنه interchromosomal shuffling during the anaphase 1. لأنه بكل cell ممكن يكون عندي different assortment of the parental chromosomes. حسب يعني مين بيكونوا بالupper pole وبالlower pole. We can have a variety يعني ما بعرف شو يعني very different, very a lot of possibilities, okay, which make that we can have the gametes are different يعني each gamete is unique and have a different constitution of chromosomes plus due to the crossing overs, okay. And regarding their role for the organism, the mitosis شو بتعمل الميتوسيس؟ growth يعني مثل we need to make more of the cells بس إنه كتر الcells but identical cells, okay. So if we had one single cell, this single cell can give me many other cells, okay, and so it will enable multicellular animal to arise from a single cell, okay, like the zygote, for example. When we have the zygote when the zygote is formed, we have one cell which is the zygote that will divide to give an embryo, okay, or yes, so to give an organism. So one cell, from one cell we can generate multicellular organisms, and we produce cells due by the mitosis. We can produce cell for growth and for reparation. Okay, for growth, as the growth of the organism needs the division of the cells to make more cells and to grow, and to repair some organs like the skin, for example, during injuries, etc. However, the meiosis is necessary for the production of gametes to reduce the number of chromosomes by the half and to introduce genetic variability among the gametes. Okay? Is it clear?
Yes.
yes.
Okay, I will stop here, and next week, we will continue.
Okay.
Thank you.